Okay, a few more things with nucleic acids and general things. One of the biggest things now is the sequencing. In other words, determining what nucleotides follow what in what order of nucleic acids. Genes, and now we got to the point where we can sequence entire genomes, whole genetic blueprints of organisms. In fact, we can do, right over in McGee Hall, we have a device that can sequence short to medium-sized sections of DNA, a few hundred to several hundred nucleotides long. So sequencing is a powerful technique. That way you can directly compare a DNA sequence from one organism to another. Or you have an organism with some kind of developmental defect, and you look at this gene involved, and you compare that gene in a normal animal, and you compare it in the one with defects and say, hey, what's wrong with this gene? Is it missing sections? Have you changed bases somewhere in the DNA sequence? Are the control sequences different from one to the other? Whatever the case is. So gene sequencing, nucleic acid sequencing, is very important. Now, there's a couple different methods for doing that. One method, I think it's the Sanger method, there's details of that in our book, lends itself to easy automation. There's details in our manual and stuff, I'm not going to go through that. But one technique is a lot more, it was done mostly with gels and stuff, but nowadays, this automated technique, there's these machines that can do this thing automatically and stuff. And like I say, the one we have in our lab, in this department, you could sequence a gene in a matter of hours, or at least a good chunk of a gene. And it's all you put stuff in, program a few things in, push the button, come back several hours later, you've got your data. So it's really kind of nice. Used to be the old days of sequencing DNA with gels and things like that. It could take weeks or even months to get the same stuff that can be done in a few hours nowadays at a relatively small regional university. I mean, like I tell a lot of people, we've got students here doing stuff that would have gotten them PhD theses 10 or 15 years ago, or if they could be done at all. That's pretty neat. Okay, so technology marches on, but the idea behind nucleic acid sequencing. Now, you can obviously sequence DNA, and you can sequence things like messenger RNAs and stuff like that. That's widely used. Now, it turns out doing these chemical tricks of sequencing, it doesn't work so well with RNA. So the best way of doing this is let's suppose you take a cell or tissue. There's techniques that allow you to extract all RNAs, separate from everything else. Other techniques that allow you to get messenger RNAs only, and all the other kinds of RNAs, the ribosomal RNAs, the tRNAs, all these different regulatory RNAs, do not get out there. So you can extract, let's say in this case, messenger RNAs. Now, sequencing an RNA directly is a painstaking and difficult process, so nobody does that. Instead, the easiest way to do it is to make what's called complementary or cDNAs. Complementary means opposite base sequence. So what we have here, here's an RNA, and I'll represent it as an RNA with a squiggly line and some bases coming off of it, some nucleotides coming off. Okay, now, what we have to do now is we have to make a DNA copy of this RNA, because DNA is a lot easier to work with. Also, RNAs are so unstable, if you look at them cross-eyed, they break down on you. They're a pain in the butt to work with. But if you get RNAs that make DNA complementary copies, then it's a lot easier to work with. And then you can sequence that in a flash. How do you do that? Well, we want to get a complementary or cDNA. So, we have to use RNA as a template to make a DNA. Now, ordinarily, we take DNA, pull a strand apart, and enzymes make an RNA, a complementary RNA. That's how we make messenger RNAs and all the other RNAs and so on. What do we call that process of using a stretch of DNA to provide the instructions to make an RNA sequence? Transcription. Okay. Well, what we're doing here is transcription in reverse. We're taking an RNA sequence and using it as a template to make a DNA sequence. So we could say this is reverse transcription. How do you do that? Well, there's enzymes that can do that. They're called reverse transcriptases. Okay, reverse transcriptase enzymes. Oh, okay. Where do you get those enzymes from? Viruses, exactly. There's a class of viruses called the retroviruses. That includes such nasties as the HIV virus that causes AIDS. And there are some others around. Retroviruses have their genetic material in the form of an RNA. But when they infect a cell, that RNA gets read like a messenger RNA, and one of the things it produces is a protein, reverse transcriptase, which then makes DNA copies of that RNA, lots of them, and then those get inserted into your genome, and then it acts like a bunch of regular genes, except it's making viral proteins, and now you get AIDS. Okay? In fact, one of those common anti-HIV medications, a class of them, are reverse transcriptase inhibitors. That alone won't stop it. You have to use several different drugs at once because the virus mutates at an incredible rate, but that's one of the tools that we use to keep AIDS in check nowadays. Okay, so for retroviruses, you get these reverse transcriptase enzymes. They can buy these commercially. So now, you take your RNAs, you make your cDNAs, and then you sequence them. And that gives you useful information. And if you have a cDNA sequence of a messenger RNA, you can make probes to that and actually get the sequence of the entire gene that codes for that. Remember we said RNAs undergo extensive processing in the nucleus before they become messenger RNAs. So the sequence of the gene has a lot of stuff that you don't find in the final messenger RNA. But it's a powerful technique. So we can do all kinds of cool things with that. Nowadays, you sequence a gene or sequence cDNA made from a messenger RNA, you publish it, and you've got to put it in one of several online databases. And they got search software in there. They got various software in there. So what you can do, and anybody with an internet connection can do this, you can type in, say, a DNA sequence. Send it to one of these databases. And within a matter of seconds, it will compare your sequence with everything else in that database and say, what are the closest matches? So let's suppose you find a new gene. You sequence it. You have no idea what that gene does, what it codes for, if anything. Type in the database, and it spits out at you what its closest matches are. And if it's something that's already well known, you can expect that this is probably a gene that codes for a particular enzyme or something else. Well, sometimes it gives you some neat surprises, though. Sometimes you find some interesting things. I'll give you an example back from my postdoc days. I wasn't the one who did this, but one of the grad students in that lab did this thing. He was looking at a protein that would take what we call microfilaments, part of the cytoskeleton, and glue them together side by side to make a bundle of them. They call these things Fondling Proteins. Okay, so it's something important in turning these fibers of the cytoskeleton into bundles, just like you stick steel rods into concrete to make like a reinforced column or something like that. So, not surprising, he purifies this thing, and then using some molecular biology, he gets cDNAs to the gene, he identifies the gene for this thing, gets cDNAs of it, sequences it. That took about a year. It can be done. Something that could probably be done in a couple of weeks right here nowadays. But that was 20 years ago. Okay, So it took about a year to get the cDNA sequence. We could do that in a couple of days, maybe a week's worth of work nowadays. So he does that, types it in a database, and the closest matches spat out was a protein called elongation factor 1 alpha. It's a protein that sticks to the ribosome temporarily and does something important in protein synthesis. So look at what's this got to do with protein synthesis? That was a surprise. Its closest match was something involved in assisting protein synthesis on the ribosome, and nothing to do with the cytoskeleton. So my postdoctoral advisor, who seemed to know just about everybody important in the field, he was an awesome person, I'll say that. But anyway, he knows somebody that's working with this very same protein. So he calls the guy up and says, hey, can you do this? Can you send us some of your protein, and we'll test to see if it glues these cytoskeleton filaments together, and we'll send you some of our protein. You test to see whether it supports protein synthesis. So this old switcheroo, and the answer was yes and yes. That turned out that protein wore two different hats. It regulated the cytoskeleton, but it also supported protein synthesis. Later they found out that it was phosphorylate, in other words, you covalently stuck phosphates on it. It did one thing, and when you remove the phosphates, it did something else. So here you had a protein with two very different cellular roles, depending on how it was regulated. So that's a kind of cool thing, but that was first found out by sequencing the gene, or at least the cDNA from the messenger RNA, and running it through the databases and seeing what was the closest match. If they didn't have that kind of technology, they just assumed it was a bundling protein and nothing else. Now we know this protein actually has two distinctly different cellular roles. So that's a cool thing about sequencing nucleic acids. Okay, well, sequencing nucleic acids is one thing, but now we've gotten to the point of sequencing entire genetic blueprints or genomes of organisms. And now we can't do that here yet, but who knows? 10 to 20 years from now, we may be able to do that. The technology is advancing that fast. One of the biggest things, the first genome sequenced were viruses. Those are a few thousand to a few tens of thousands of nucleotides. And then bacteria, usually a few to several thousand bases long for their DNA. And then finally, yeast, which is a little bit bigger, about 10 million. And then finally, the first multicellular organism that underwent development. It's a little tiny roundworm named C. elegans. Okay, this guy has 250 million pairs of bases in its DNA. Took many years to get that sequence. And then that big, huge human genome project in the 90s, the idea was to sequence the entire genetic blueprint of one or more people. When they first proposed it, it was looking like it was likely to take a couple of decades and cost $20 or more billion. But technology marched on. They got it much ahead of time, in less than 10 years, and the whole thing cost about $1 or $2 billion. One of the rare cases where a government project actually cost far less and takes far less time than you originally anticipated. The sequencing technology has gotten so advanced now that there are companies that have devices that can sequence a human genome for $5,000 to $10,000. The holy grail is to drop the cost of a genome sequence to $1,000 or less. Because when that happens, and that should be within 5 years, when that happens, you go to your doctor's office, and one of the first things they're going to do is draw a little blood from you, send it off to a sequencing lab, get your entire genetic blueprint, and look for markers that indicate increased risks of this type of disease, that type of disease, or maybe that this drug will work fine for most people but can give you really nasty side effects if you use it. And once in a while that happens. You hear these things, there's been a number of cases of drugs being FDA approved after these clinical trials involving a few to several hundred people. Looks good, looks safe, looks effective, then goes in the broader market. Now tens of thousands, even millions of people use it, and some of them get killed by it. And then they pull it back off the market. That's happened. Once in a while, you have these rare mutations or whatever, or a particular drug that works fine for the vast majority of people, has dangerous or even lethal side effects for others. So there's been several cases where they put drugs on the market, and a couple years later, when the body count started rising, they pulled it back off again because extremely rare but extremely nasty side effects, like death. Death is a pretty good nasty side effect, right? Okay, so.
I have a question.
Yeah.
In genomic sequencing, you know, Alzheimer's wasn't on my side of the family, but they told him a few years ago they could test to see if he was a carrier. Do they do that way? Is that what they're testing for?
They rather than whole genomes, they would get probes against specific mutations in a particular gene that can run in the place. There's probably one thing, this is called ApoE4, is that the one they're looking at?
I think so. Okay,
that one seems to be connected with a, if you're a carrier, heterozygote, you have about 4 or 5 times higher chance. If you're homozygote for that variant version, you've got about 20 or 30 times higher risk. Yeah. So so that is about the one gene that we know of that's really strongly correlated with Alzheimer's. There's probably scores of other genes that people are looking at that, but haven't gotten there yet. But yeah, so they would use a marker, and this is your standard genetic testing for this cystic fibrosis, a probe in effect that would look for a specific mutation. And you got it or you don't have it. In my case, they used a probe that was looking for a particular gene that's very important in regulating cell division in my lymphocytes, and lo and behold, they didn't find it. That is a very bad thing. Okay, so see, I'm missing this one. In fact, I'm missing 2 genes that are very important in regulating cell division, so these cells are dividing out of control. So those kinds of things are easily done now. And they use the hybridization technique. The same things that we described. So this stuff is also not just in the research lab, it's used in a lot of clinical labs now. Okay, so sequencing genomes, that's very important. Of course, you can compare the genomes of one organism to another, and we find some remarkable things. One of them is many critical genes involved in development are very similar even from vastly divergent organisms. Most of the genes found in that roundworm, we have equivalents ourselves. Same thing with fruit flies. Many of the important developmental genes, we've got equivalents. In fact, we often have many different versions where fruit flies may have one gene, we may have a dozen. We're more complex organisms than fruit flies. And so you can do a great deal. That's how we found out that genetically we're 98% similar to, it's identical to chimpanzees, and even less in the parts of the DNA that code for, that have the instructions for protein coding genes. That's even less. The difference between chimpanzees and us is not so much our genes are different, it's the regulation of certain genes, especially ones involved in brain development. That's what's different. Okay.